Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome everybody again to another episode of our show. Today bringing you another really awesome guest, talking about a really interesting area of drug development. Today we have the honor of being joined by Dr. Ryan Potts, who's Vice President and Head of the Induced Proximity Platform at Amgen, which is a drug development strategy. It's focused on novel ways to bring together two or more molecules in close proximity to one another to ultimately tackle drug targets that are currently considered undruggable. And we'll get a little more into what that means in the episode. Dr. Potts also leads Amgen's Research and Development Postdoc Fellows Program. He obtained his bachelor's in biology from the University of North Carolina, followed that up with his Ph.D. in cell and molecular biology from UT Southwestern in 2007. 2008, he was awarded the Sara and Frank McKnight Jr. faculty position at UT Southwestern Medical Center. And during that time, his lab was focusing on answering longstanding questions in cancer biology, specifically focused on cellular function of cancer-testis antigens, proteins. In 2011, he was appointed assistant professor in the Department of Physiology, Pharmacology, and Biochemistry at UT Southwestern, and his work there to find an interesting function for the MAGE gene, or the Melanoma Antigen Gene. Family of protein regulation. 2016, his lab moved to St. Jude Children's Hospital, became an associate member of the Department of Cell and Molecular Biology. There, his team continued to work on CTAs, as well as focusing on biochemical, cellular, physiological, and pathological function of the MAGE gene family. And ultimately, he moved in 2020 to Amgen to create this entirely new department focused on induced proximity. And really excited to have him with us. A lot of interesting topics. I'm looking forward to the discussion. Dr. Ryan Potts, thank you so much for taking time to come on the show today.
Yeah, thank you so much, Ira, for the kind invitation, and looking forward to chatting.
Yeah, absolutely. I'd love to start off, as we typically do, by going back to the beginning. In your case, I read all about you. Your interest in STEM and biology and cellular molecular biology started quite early in life, and I was wondering if you could sort of tell us the background story to your background story, because I think it happened when you were 2, that sort of the world of biology and cancer biology and everything came crashing down upon you. Love to hear your background story of what got you ultimately into the healthcare space and focused on this really interesting area.
Thanks for the question. I think that is a fairly unique way to sort of get introduced into the area of biomedical research and drug discovery. As you mentioned, at a very early age of 2 years old, I unfortunately was diagnosed with a brain tumor that was really impacting a lot of the core sort of endocrine functions, particularly the pituitary gland, and it required surgery, intervention, but also meant that a lot of the sort of normal functions that the pituitary does, I was left without. And so it became sort of, life became dependent on medicine that, I still take today. A number of sort of supplemental hormones and things like that you take for granted, I sort of required to take synthetically. And, that really sort of experience of sort of being around healthcare, being sort of in and out of the hospital. Unfortunately, the tumor reoccurred when I was 9 years old. And of course, at that age, then I can start to remember all the experiences associated with that and sort of start to ask the questions of, why does this happen, right? And, um, how does the body respond to these types of medicines, and how do they work? And I think I developed a very big drive in me of understanding and asking the question why, and that led me, I think originally around a career path of thinking that I would go into medicine because I'd obviously had been around the healthcare and had a number of physicians over my childhood that I'm real grateful for because there's been a lot of effort getting me to where I am today, and through that challenge. But as I went to college, at UNC Chapel Hill, as you mentioned. That's also where I had all my treatment for when I was a small kid dealing with the brain tumor and the sort of chronic conditions associated with that, and got the opportunity to actually work with the pediatric endocrinologist that who had been treating me for now at that point almost 18 years. And he was a physician scientist that had a laboratory, and so my freshman year at college, right out of the gate, I started working in his lab. Originally, this was just sort of the typical things that what undergrads do. They wash the dishes, they restock and supply things and whatever. But over time, got more and more sort of training around molecular biology and biochemistry type techniques, and it just really, I got really excited and interested in sort of, again, how things work, and that that led me to then make a decision to pursue a career in research after graduating there with a bachelor's in biology.
I appreciate sharing that part of the story because it ties in obviously very well and connected to where we're going with this. But before we get into everything that we're going to be discussing about your current portfolio in terms of reduced proximity to Amgen, I wanted to stop at a couple of other places because a lot of the drug development that we're going to be hearing about that you're doing now is not just novel, but you're dealing with really interesting biochemical moieties that do for lack of more than one thing. And I think we see that in a lot of your work pre-Amgen. A couple interesting places. One I'd love to stop is just at your dissertation, where you worked with these really interesting SMCs, structural maintenance of chromosome complexes, which again, don't do one thing, but are, in the best description I can put it, there are these really interesting little motors that use other motors like cohesin and condensing complexes to protect our genome, to help it grow, to protect from genotoxic responses. Not very simple compounds we normally learn about in biology, but really complex activity happening. Talk a little bit about your dissertation, if you would, and a little bit about sort of what this taught you about the complexity of what's going on in the cell, let's put it that way.
I think you're dead on, but one of the early sort of learnings during my time, it actually started a little bit before my dissertation. Before joining a PhD program at UT Southwestern, I actually stayed on at UNC Chapel Hill as a research technician for about 2.5 years, working in a neuroscience lab, and that helped lay a lot of this foundation. But I think the key thing that I took away from a lot of this is just around, as you mentioned, the complexity of biology, and that understanding the fundamentals of how cells work and the biochemical reactions that drive everything that's happening in a cell just fascinated me, and is really important for what we do in terms of now with drug discovery. in my dissertation, I was curious to pursue something that was quite novel. And at the time, as you mentioned, this complex known as the SMC5/6 complex was just sort of starting to be identified, but there was really no known function to what this complex of proteins did. As you mentioned, they're related to these other proteins known as cohesin and condensin. They're all sort of protein complexes that modulate the shape and assembly of chromosomes in different ways. Cohesin is very important in mitosis, as well as condensin, and cohesin mediates this sort of holding together of sister chromosomes to allow proper equal segregation to daughter cells during cell division. Yep. Condensin is also important in that same process, but it condenses down the DNA that's like, super long in its linear form. And obviously it needs to be compacted down to properly segregate to daughter cells equally. But there was this new complex that was related called SMC5/6 that we really didn't know what it did. And my thesis was really about understanding its function. And we quickly linked it to involvement in DNA repair and further uncovered that it's important for homologous recombination. And then we started thinking about how this was involved in disease. And it turned out that homologous recombination is actually quite important for maintaining the proliferative capacity of a certain type of cancer cells where the ends of the chromosomes known as telomeres undergo this sort of shortening event with ever proliferation. So cancer cells have to find a way to elongate telomeres to maintain this sort of limitless replicative potential. And a fraction of cancer types will utilize a recombination mechanism where they elongate as opposed to sort of the natural mechanism of the telomerase enzyme. And so SMC5/6 complex is really important in this telomere recombination to maintain the length and allow the replicative potential of cancer cells. Mm-hmm. So, a lot of novel biology, novel sort of biochemistry, understanding post-translational modifications, including things like simulation. And, I think the one thing that I really took away from this time that stuck with me for a long time is really trying to think about science in a way that we're not just doing incremental science. I'm sort of not interested in doing sort of the +1 type of science. I'm interested in sort of, emerging from the herd and, going after the most impactful work that we can. And I feel like oftentimes we get too sort of called up into sort of, I would say, like more of this lamppost effect where everybody sort of searches around where the light is. Right. And I think there's a lot of interesting biology in application to drug discovery off in the dark space. And that's what I feel like my thesis helped me recognize and sort of set up my future career path with that sort of knowledge in hand.
And a couple of years after that, you and one of the papers is the Comprehensive Guide to the MAGE family of ubiquitin ligases. This is Journal of Molecular Biology in 2017. Again, we see this in your work in the sense, as I mentioned, the bio start with the melanoma antigen genes. Melanoma, yeah, but it's not much more than melanoma. It's potentially, many cancers, as well as neurodevelopmental diseases, renal and lung disorders. And attached to that is, and we'll be getting this in a bit, in this case, the whole process of ubiquitination, a wonderful sort of post-translational mechanism that does all sorts of things in terms of degrading, trafficking, and it can be used if properly, controlled for many other possible therapeutic applications. Say a couple of words about this work as well, and then we'll be getting into the principle of undruggability.
Yeah, thanks for that question too. This is really where I think my interest has started to solidify around sort of molecular mechanisms regulating cellular processes in the cell. And you mentioned around ubiquitination and protein degradation. So it was sort of as I was completing my thesis that I recognized that there was this protein within the SMC5/6 complex that we were just talking about that had this domain, a protein domain that I had never heard of. And it had this domain called a MAGE homology domain. And that sort of piqued my interest because sort of like there's, domains are fairly common and most of them sort of have ascribed functions. And this one, there was no ascribed function. And so as we talked about, I'm sort of interested in exploring the unknown. And this was a case where it was like, oh, this is really interesting. It turned out there's a whole family of these proteins. As you mentioned, the melanoma antigen genes that all have this conserved MAGE homology domain. And as I dug into this even further, they have this very intriguing expression pattern that the majority of these are only expressed in reproductive tissues like in the testes, but they're turned on aberrantly in the context of cancer and many cancers, including melanoma, where they were originally discovered, and that's how they got the name, but they're turned on in many different cancers. And there's probably at least 50 different MAGE proteins in the human genome. And very, very little information of what they actually did. And so I was at the point where I was transitioning from my thesis work, and I had the opportunity to sort of skip the postdoc phase and join as a junior faculty at UT Southwestern. So I was looking for sort of what was I going to do to sort of launch my career. And I felt like this was something that like checked all the boxes, where there was a really interesting question. There was a lot of unknown, but there was a big impact that could potentially be had to understanding these the function of these proteins. Yep. And so I launched sort of my independent career around essentially deorphanizing what these proteins did. Yep. And that led into a number of different biochemical, biophysical, structural, physiology, pathology types of studies that over the course of 10 or 12 years uncovered the function of the MAGE protein family is that there are ubiquitin ligases. They bind to these proteins in cells and facilitate ubiquitination of targets to essentially rewire the proteome. So you can think about this in the context of cancer, where most cancer cells don't express these genes, and then suddenly when the cell becomes transformed and diseased, these genes turn on. The genome is messed up, and the MAGE genes are turned on, and they turn on, and then they start promoting the degradation of specific proteins like tumor suppressors. And that sort of is a kickstarter of rewiring the proteome into a more of a state that's conducive to the sort of properties that we know of tumorigenic cells like proliferation, et cetera. So that sort of really got where I invested a lot of time in my independent career, and I think was very rewarding to make that discovery that sort of launched a field around this MAGE gene family.
Excellent. I appreciate you going into both of those bodies of work because they converge on where we're going here. But so we're here we are in 2024, 20 years since the completion of the Human Genome Project. Tremendous amount of data comes out of that. You mentioned the proteome as well, transcriptome, a lot of other multi-omics activity happening in our industry. The world thinks about we have thousands and thousands of targets that are going to keep the pharma industry busy for the next, couple hundred years. But no, the story is not that simple. And unfortunately, the fact is, as you point out in all of your work, upwards of 85% of a lot of these targets, in terms of proteins, other molecules, are considered undruggable. And I'd love you to take a little time to go into what exactly that means.
Yeah, it's a good question, and I think we actually should start thinking about maybe not undruggable, but maybe difficult to drug, because what
Okay.
we're doing is really trying to make that term obsolete. But let's think about this from a standpoint of what is difficult to drug. Many proteins in the body really don't possess the kind of functional pockets or crevices where a therapeutic molecule can bind effectively and alter the protein function in a way to change the course of a disease. Think about this in terms of like a lock and key. So most proteins that have been drugged by sort of conventional mechanisms and molecules for the last 100+ years, those proteins are sort of like a lock, and they have a little keyhole. And if you have just the right key that has just the right shape to it, you can stick it in that keyhole and you can sort of unlock it. You can sort of make the lock useless, right? Because it opens up. Well, that's sort of how the proteins have been drugged in the past. They have the proteins that are amenable by these conventional methods have these sort of keyholes, and the molecules, the medicines, are discovered and designed to fit into those keyholes and sort of turn off the protein. And that works great when there is such a keyhole there that you can do that. But the reality is that probably 85% of proteins encoded within the human genome don't have these kind of keyholes. And so we got to think about ways that we can develop new types of molecules and new types of mechanisms to control these proteins that we'll call difficult to drug, by novel types of mechanisms and modalities. And that's really where we're focused, as we believe that we're entering a new wave around drug discovery innovation, utilizing this concept of induced proximity that you started the show with, as well as this what we call multi-specific molecules that really pave the way for new types of treatments aimed at the thousands and thousands of disease targets that are currently considered difficult to drug.
And is it, again, for everyone listening, so we're not, I take my SSRI and I boost my serotonin. I take my cholesterol blocking drug and my cholesterol goes down. We're well past that, where we're not focusing on the keyholes anymore. And as you were mentioning, we're talking about the induced proximity platform and this principle of a multi-specific molecule, something that ultimately, and the way you describe it, and there's an awesome video by Amgen that I encourage everyone to go look at. Well, there's two actually, one with Dr. Potts and another awesome cartoon, and we'll put links to those in the bio. But in essence, these very novel molecules, which I'd love you to describe, and we'll go into a little more detail of the individual ones, that basically act as so-called molecular matchmakers that, in the case of the little cartoon, hey, Mr. Misfolded Protein, hey, Mr. Proteasome, I'd like you to meet, and for nature to take its course. Walk us through a little bit of how you develop the IPP and what a multi-specific platform concept, and just a little bit about, if you can, just what it was like initially setting up this entirely new novel concept at an organization like Amgen.
Yeah, thanks for that question. And yeah, the cartoon is fabulous. It really puts everything in perspective that is easy to sort of understand, even for folks that are not focused in this kind of work all the time. I think maybe it's worth just setting up the historically where we came from and to get to today, right? And we think that you can sort of define that there's been probably three major waves of innovation in drug discovery over the last century plus that impact modern medicine. I think that started at the early 20th century in the initial wave, where you had medicines that like worked, but we didn't really understand why they worked. And aspirin maybe was a good example of that. We have these well-defined chemical structures, but the really unknown biology. And then, that was sort of status quo for a very long time until maybe like in the 1970s or so, you started to have the second wave where it was more like rational drug design, where we had specific compounds that were aimed at specific molecular targets that we then sort of understood and knew played a role in disease. And that was sort of a major breakthrough. And then that gave way to maybe the third wave in the 1980s, which was driven largely by advances in recombinant DNA technology, this sort of biotech revolution that came from that. It allowed us to go beyond chemical compounds as drugs to actual proteins and other types of biologics. And that's really been a boom for patients that impacted through diseases where we can govern biology through these large molecules. Now, I think we're entering this wave 4. And we believe this is sort of characterized by this concept of multi-specific medicines. And this is really about, it's quite radical in changing how we think about conceptually of drugging targets. And we just talked about sort of the conventional way of the lock and key, but multi-specifics don't work like that at all. Right. There's no keyhole where if you stick a molecule in there and that's going to do something. Instead, as you brought up, multi-specifics can harness the sort of power of cell biology using this concept of molecular matchmakers to link a disease-causing target to a potent natural mechanism that act upon that target. And I mentioned earlier that maybe 80% or more proteins are sort of difficult to drug by conventional mechanisms or drug modalities, but of course Mother Nature drugs 100% of things, right? 100% of the things that are produced in the body are regulated by the body in a natural sort of mechanism. So if there are machinery in the body that can take care of these things naturally, why can't we use molecules that engage that machinery and sort of give it new directions of, go address this protein over here because it's causing a disease? And that's what really multi-specifics are about, induced proximity, to identify these kinds of molecules that induce the proximity between a disease target and some type of natural cellular mechanism to allow you to modulate that target in a predictable and desirable way to have impact on the course of disease and ultimately for the benefit of patients.
And you wrote a review in Chem Biochem in February of this year entitled Tactics for Leveraging Proximity Biology and Drug Discovery. And in the review, you basically summarize sort of two baskets, and I'd like to go into both of them. One, focusing on a strategy of induced proximity related to the degrading functions of our cells, and then to non-degrading modalities. And I think we could talk about both of these. And I think an interesting place to go first would be something that you published quite a bit on in the last couple of years, and the principle of the concept of PROTAC, or proteolysis targeting chimeras. In this particular case, again, I'm simplifying this, our bad guys here are unwanted damaged proteins. Our good guys are all the interesting degrading machines that we have floating around in our cells, the proteasomes, the lysosomes, and so forth. We want to bring them together. And in the concept of PROTAC, you created what you define as a heterobifunctional molecule to do this. Take a little time and walk us through exactly what this means. And obviously, targets that come to mind are things like obviously Alzheimer's, where we have poorly folded proteins that are doing real bad things in our brain. But if you want to give examples of other really interesting targets as well, please.
Yeah, I think you described it quite well on what PROTACs are. These, as you refer to, proteolysis targeting chimeric molecules, as they were initially named, acting like molecular matchmakers. They're typically molecules that sort of have two sides to them. They're sort of bispecific or dual binding molecules, where one end of the sort of barbell shaped molecule will bind to that protein target, that disease protein that you want to modulate and in this case remove from the cell. And you mentioned that certain types of targets that might be of interest, you included things like damaged proteins. I think that's very interesting, right? Because they're likely mucking up the system, and you want to just get rid of them. There's a lot of opportunity in the neuroscience space with damaged proteins that accumulate over time that you like to clear, and protein degradation is a good opportunity to do that. But there's many other proteins that you like to remove from cells, and it includes many of these we've just been talking about. They're just sort of classically difficult to drug because they don't have these sort of pockets with them where molecules can fit and sort of inactivate them. And so you got to leverage the degradation machinery that the cell has evolved over many millions of years to leverage that and to degrade these proteins. So you have a molecule that binds to the disease target, and then it is linked to a molecule that binds to one of these degradation machines in the cell. We call these ubiquitin ligases. Mm-hmm. These are a specific class of proteins, and they have the job essentially to mark the proteins that should be degraded. It's almost like sort of maybe actually in the cartoon, it's the little traffic cop that goes around writing tickets, and you stick that on a car, and well, then what comes next? Well, the tow truck comes along and like gets rid of it, right? In this case, the ubiquitin ligase is like the little person ticketing something, and what it does is it marks the protein physically. It puts an additional small protein on that target. That small protein is called ubiquitin, and that ubiquitin is a signal to the cell that this protein needs to be removed. And so it is then recognized and dragged into a sort of molecular machine that sort of is like a barrel, and inside the barrel are sort of like molecular scissors that chop up that protein and therefore remove it from the cell. That machine is called the proteasome. Mm-hmm. And so these PROTACs, they induce the proximity between the disease target and the ubiquitin ligase, allowing the ligase to modify the target, and then that is a signal to be recognized and destroyed by the cell's degradation machinery. So there's many opportunities around leveraging these kinds of molecules, and we're really excited about the opportunity for targeted protein degradation, as we call it, in the field. And it was originally sort of ideated by Ray Deshays and Craig Cruz, a number of years ago, maybe back in the late 90s, around 1998, I think. Just sort of hatched over beers on a napkin, this concept, and collaborating together, they developed the first sort of proof of concept of PROTACs that gave way to this huge field and this huge opportunity to make medicines to treat diseases with high unmet need that we haven't been able to in the past. So I'm really excited about this. Within the induced proximity platform, we study targeted protein degradation here at Amgen, and utilize and design and discover PROTACs that can work with different types of ubiquitin ligases. Mm-hmm. There's related kind of molecules that we can talk about later if there's interest. We call molecular glues.
Sure.
But this is a really interesting set of work. And, I'll just give maybe one example of a program that is sort of actively ongoing, both within the induced proximity platform, but also ongoing within the field as a whole, that sort of highlights the potential of induced proximity and targeted protein degradation using molecules like PROTACs, and that is around treating a form of lung cancer that has remained very challenging to target, where there is a mutation in a protein called SMARCA4. And what ultimately that yields is that the cells become particularly vulnerable because there's a not near identical twin protein to SMARCA4 called SMARCA2. Okay. And so most normal cells have both SMARCA2 and 4. However, these tumor cells, they have mutations in SMARCA4, so they're only left with SMARCA2. And it's been known that that's a sort of context of vulnerability, because if you remove the remaining copy of SMARCA2, these tumor cells are toast. They can't live without any SMARCA2 or 4. And but it's been challenging to drug that because the SMARCA2 and 4 are so essential and they're so similar to one another. Any kind of inhibitors have been sort of hit both SMARCA2 and 4, and that's sort of toxic to all your cells in your body. Mm-hmm. You got to design molecules that only touch and modulate SMARCA2, but not SMARCA4. Okay. And that's been where the opportunity comes from induced proximity, because now, since you're utilizing these sort of cellular machines in the cell, and in particular ubiquitin ligases, you can design molecules that selectively target SMARCA2 with that ubiquitin tag and remove it without affecting SMARCA4. So this is just one example of how in the past we've sort of been unable to approach this target and impact this disease. But now we've been designed PROTACs that selectively target SMARCA2 for degradation, block tumor cell growth, and still sparing normal cells.
Excellent. I also love the barroom napkin part of it as well. But no, that's an excellent example. Again, thinking about the oncology space, but that obviously there's so many diseases that are based on these misfolded proteins that one can see how you could create. And again, you were talking about how many things the ubiquitin ligases can impact, that you can mix and match in terms of these matchmaker systems that and come up with quite a wide range of these bifunctional opportunities. So that's exciting. On the other side of the that recent paper was the principle of the non-degrading modalities, and I thought this was an interesting time maybe to talk a little about RNATACs. And here again, we're talking there's many different types of RNA out there that we are familiar with, that we've talked a little bit about on the show in terms of mRNA and microRNAs and small interfering RNAs and so forth, all extremely important in biological processes. Again, in this concept that you recently wrote about a month ago in Cell Chemical Biology, the bad guy is, again, well, not really bad, but an RNA that we want to impact in some way, difficult to target with traditional drugs. You're bringing the induced proximity concept in bringing things like RNases, ribonucleases, to alter not the destruction but the stability, how something is ultimately spliced, translated. I know it's a little early in the process, but can you say a few words about your vision for RNATACs?
Yeah, thanks for that question. And we are really excited about induced proximity mechanisms beyond targeted protein degradation. It all started with targeted protein degradation, and that is still a major focus for sort of induced proximity for drug discovery. And it will allow us to get at many difficult to drug targets, but it probably will not let us get at everything. And so we sort of have this this mentality here at Amgen that, the sort of tagline of any target every time.
Mm-hmm.
And so we want to sort of be prepared when we have sort of high conviction targets that, we really believe are, gonna impact the course of disease and are gonna help patients in need. We want to have the ability to go after those targets, and no matter what the mechanism, no matter what the modality might be. And we believe induced proximity gives you many more options to do that. And we talked about that for targeted protein degradation, but there's going to be certain types of targets, proteins in the cell that are just not amenable to that approach. Just to give you maybe an example or two.
Sure.
That degradation machinery I described, it's not everywhere in the cell. Like, for example, well, it's, for example, not outside the cell. Many proteins are secreted. So you're not going to be able to approach those with the targeted protein degradation by the conventional means that we described earlier. Or some proteins that are in certain organelles like mitochondria. There's no, ubiquitinases and proteasomes in the mitochondria. Some proteins don't have the types of amino acids like lysines on their surface where that ubiquitin tag gets conjugated to. And so that they may be challenging to or impossible to drug through the targeted protein degradation effort. Even some of these misfolded proteins that you alluded to as being important in the context of neurodegenerative diseases, proteasome can have a hard time. As I mentioned, it's sort of like this barrel, and you got to thread the protein down to a very small hole into this barrel to get chopped up into little pieces. Well, a big sort of aggregated protein that's like a big ball of yarn that I don't know, maybe it's sort of like those Christmas lights at the end of the year where it's just like they're all strung together and it's almost impossible to detangle it. Well, it's sort of like hard to get these aggregated proteins to get detangled down into the proteasome to get degraded, right? So you got to have other ways to get at targets beyond targeted protein degradation. And induced proximity allows one to do that in many ways. And one way that we think is interesting, but it's very early and is a very challenging approach, is that if you can't approach the protein, then of course all proteins come from RNA. And so if you can modulate the RNA, you're effectively able to modulate the protein. Right. And then of course some genes don't code for proteins; they just code for RNAs that have function in and of themselves, right? And so that's another opportunity with the concept of modulating RNA by induced proximity. You can get at these RNAs that are not protein coding. So we are sort of developing a concept around induced proximity where we have molecules that again redirect the natural cellular machinery that is there evolved to control RNA stability or control its splicing or its localization or translation, whatever it might be. But now we have molecules that can redirect that to disease targets of interest. Mm-hmm. And so the same concept that we just talked about on sort of molecule design fits here, where one end would bind to the disease-associated RNA, and one end would bind to a cellular effector mechanism like an RNA-modifying enzyme. And that would bring the RNA-modifying enzyme, like a molecular matchmaker, to the RNA target and modulate the RNA in a desirable way. Mm-hmm. So this is just I think one example that shows, illustrates how induced proximity can be applied in many different ways to have many different outcomes and expand sort of how much of the target tree that we can really address. Mm-hmm.
Mm-hmm. And you mentioned before the topic of molecular glues, and in that case, you're, and correct me if I'm wrong, you're not really dealing with putting something in proximity with a machine of any type, but really two proteins that together have some type of synergistic benefit. It got me thinking back to your PhD about these complex
Yeah.
proteins, moieties that do important things for us. Could you say a couple words about molecular glues while we're on this topic?
Yeah, I think that's a good analogy. Many proteins basically work like a molecular glue, but like a really big one.
Right.
Their goal is to sort of bridge together, bring together two other proteins so that some type of reaction can occur. I mentioned that in case of like the MAGE genes, they're sort of like a molecular glue. What they do
Sure.
is they bring novel substrates to ubiquitination to allow the degradation and revamping of the cellular proteome. Um, Molecular glues, in the context of drug discovery, are essentially small molecule versions of that. They bring together two proteins and sort of stick them together. They have the sort of subtle variation from things like we talked about with PROTACs or RNATACs in the concept that those are sort of bifunctional molecules
Sure.
where they have high affinity binders on either end, one binding to the target, one binding to a cellular effector. Molecular glues are sort of more streamlined molecules, more compact. They typically don't have high affinity binding parts to both sides of the complex of the target and the effector. Rather, they typically interact with more higher affinity to either the target or the effector.
Okay.
And then they mediate interaction to novel substrates or novel proteins through forming this ternary complexes. So they're very interesting because they're typically more drug-like. So you can imagine like this barbell type molecule I described earlier is not sort of um, conventional in nature. So you need a lot of like optimization of that to make
Sure.
it into oral medicine. Whereas a molecular glue is more streamlined, it's more compact, it's more consistent with how molecules that are drugs sort of look like. But the challenge has been how to discover those in a prospective way.
Right.
Most of all the molecular glues, particularly the degrading type of molecular glues that have been discovered sort of serendipitously, where there was some type of phenotype that was screened for, and in the end, it was realized that they actually worked as a molecular glue degrader, degrading some type of target, leading to some type of phenotype. And so we've really put a lot of effort in induced proximity group here of how do we actually do molecular glue discovery in a prospective and a rational way. And we've invested into um, building out sort of chemical libraries that are highly enriched for molecular glues.
Mm.
Sort of like turning the concept of finding the needle in the haystack on its head,
Right.
because these molecules are so rare, it's often been referred to as sort of because they come through this serendipitous way of sort of finding the needle in the haystack. But we believe that, it's much easier to find the needle if the haystack is just full of needles, and that's all the haystack is. And so we build these sort of custom chemical libraries that are enriched for these molecular glues that allow us to do this in a much more rational way. And so we're pretty excited about that.
No, definitely, definitely. Right, as we're talking a lot about the inside of the cell, and a lot of this just got me thinking. We've done several immuno-oncology episodes, obviously on these interesting biologics to target cancer cells. But we've also talked about, these sort of, again, these natural processes like cellular competition that, throughout our life, for the most part, keep us sort of cancer-free when things go wrong with cell A, other cells in the proximity of that cell, nudge it to apoptose and do other things. Any interesting applications of induced proximity and some of these concepts that you're talking about for, the cell in general, and how we can potentially bring together our sort of natural, tumor suppressant capability or, oncolytic capabilities to destroy entire cells.
Oh yeah.
Or am I talking outside of
No,
my
no, no, no.
capabilities here?
I think it's a great suggestion and sort of thought there. Um, yeah, largely what we've been talking about up till now has sort of been like modulating the cell from the inside, right?
Right,
And
right,
so, um, can you modulate the cell in ways that is desirable from the outside? And that could include, as you described, of like just eliminating cells altogether
yeah,
that are diseased. And there's definitely some good examples there around induced proximity I'll get to just one second. But of course, it also could be um, modulating cellular behavior through controlling the cell surface home, right? There's a lot of signaling that happens at the cell surface. So let's go back to your first example, and that is around eliminating, say, tumor cells.
right,
And I think a good example of that that really relies on this concept of induced proximity that Amgen has has been a real leader around is around bispecific T cell engagers.
okay,
These are typically large molecule proteins that have sort of multiple different components to them. That one component binds to the cancer cell, recognizes some type of cell surface marker that is specifically on the cancer cell. And then it's fused to another protein that binds to a T cell, an immune cell whose job it is to destroy other
right,
cells in the body that are bad.
right,
And so by bringing that T cell in proximity to the cancer cell, you can elicit a T cell response that ultimately kills that tumor cell. So that's sort of a fits sort of your idea of can you modulate cellular behavior, or in this case, just promote cell death through induced proximity between two cells on the outside.
that's a fascinating example. And again, I get I've always been sort of focused a lot on not just the oncology space, but a lot of what's happening in regenerative medicine and sort of how we need to harness not just the ability to regenerate stuff, but also to properly control, say, senescence, or when we get
Yeah.
into more complex tissues, we want certain tissues to grow, other tissues not. So I think obviously not being an expert in the space, it seems like you have a really broad potential here with the platform, and I'm just super excited about it personally. So no, that's really wonderful.
Yeah,
Um,
I'm really excited too about these opportunities. I mean, they're just it's a bit of a creative playground for any scientist to innovate around and to come up with ideas that you wouldn't have dreamed of before. But
right.
now with this sort of concept of induced proximity and making these sort of multi-specific molecules that have really transformed what we can do. And so it's a really exciting time to be in drug discovery, and the future is bright.
Yeah. And you mentioned a creative playground. One of the things when we talk with groups like the DARPAs of the world, this principle that it's nice that, especially with a company like Amgen, it's letting you think outside the box about these things. So we only think about in the small biotech companies, but the fact that here you are leading this important group at Amgen is really impressive, and it's exciting to see that happening inside. Um, what else is happening? I noticed a couple weeks ago there was a Gordon Research Conference on molecular mechanisms of protein processing, trafficking, and secretion. There you were talking about PROTAC. What else is coming up for the year? Obviously, you got a lot going on internally with the programs, but other conferences, talks, other external initiatives that you're going to be involved in spreading the message here. What else is coming up that we should know about?
Yeah, good question. I'm a little worried you're too close on what my travel schedule is like there, but no, there's a lot of great things happening in the field, and as you mentioned, because it is such a um, rapidly evolving field, you got to really stay connected, and each time we have these sort of major conferences in this area. Just amazing what work is going on both in the sort of academic space as well as in the drug discovery industry, biotech space. For me, I'm really excited about some things that are coming along. There's a couple of new um, targeted mechanisms of induced proximity that we're going to be publishing in the second half of this year. So I would watch out for those, and they're sort of quite creative and non-conventional. And some of these are actually led by postdocs that are here at Amgen.
Yep.
I lead the R&D postdoc program here, and so
Right.
that's something that's really exciting as well. So stay tuned on that. New mechanisms to control protein degradation beyond the proteasome and ligases that we just talked about. So there's a couple of cool stories coming.
Awesome. Awesome. And yeah, I apologize. 'Cause I did mention that in the bio, but could you say a couple more words about, because you also, in addition to all this, you do lead this Amgen R&D
Oh yeah.
postdoc. I know it's active in the United States, Canada, and Denmark, I think, currently. And just can you say a couple words about any aspiring postdocs that are going to be listening, and what's involved here? Because I clearly that's an interesting way for the next generation to get involved in what's happening at Amgen.
Thanks for the question and the opportunity to mention that. Yeah, a lot of I think a lot of students don't actually realize that you can do a postdoc in industry. I actually didn't have much vision into that myself before moving to Amgen. But yeah, we run a global postdoctoral fellows program at Amgen, where it's designed around training sort of the next generation of scientists, primarily that want to go into drug discovery in a biotech and pharma setting, but not exclusively. We have folks that do other things after the end of their postdoc. But it allows trainees to come into Amgen, be integrated into what we're doing, come up with creative projects, all designed around being publishable and being led by the postdoc sort of intellectually. And it's just a great opportunity to get exposure to how industry science works, how they can get involved and establish a career in doing really just outstanding science within the drug discovery industry. So yeah, I'd recommend anyone that's interested in that to look into their industry postdocs like the Amgen one. They're typically around 3 to 4 years, and it sets you up well for your next step sort of in your career development towards a job in industry.
Awesome. Awesome. Really a great story, Ryan. I appreciate you going into everything you're up to, both with the IPP program as well as the postdoc work. Wishing you and the team the best with all of it as you continue to move it forward in 2024 and 2025. And I hope to do a follow-up with you down the line as things progress. Again, for everyone that's going to be listening to our show across the various podcast networks, or who will be watching on our YouTube channel. Again, you've been listening to Dr. Ryan Potts, Vice President and Head of Induced Proximity Platform at Amgen, also lead for Amgen's R&D Postdoc Fellows Program. Ryan, really appreciate the time you spent talking about what you're up to, telling us your story. Thanks for everything you obviously do for patients. And as we like to say on our show, thanks for helping to create a better tomorrow for so many people out there via what you're doing. Really a great story.
Yeah, thanks so much, Ira. It was a lot of fun, and look forward to following up in the future.
Will do.